Hello. Hello, is my voice audible?
Yes, you're audible.
Good evening, everyone. And we will begin the program shortly at 6:45 p.m.
চিৰন্তন হেল্ল' চিৰন্তন আছানে?
Hello, sir.
আছা চিৰন্তন?
Sir, Sirantan join কৰিছে, sir.
মই জইন কৰিছো তাকে জনাবলৈ
yes sir মই আছো sir মই হেৰি লগত কন্টাক্ট কৰি আছো
ঠিক আছে মই ভিডিঅটো জাস্ট হেৰি কৰি থৈছো মই আছো মোক কৈ দিবা
yes sir হয় sir মই কৈ দিম
Hi Maina. Hi Maina.
দিগন্ত
Maina.
আপোনাৰ হেৰি লেটাৰ পালো দেই থেংক ইউ
ঠিক আছে
থেংক ইউ হেৰি আপোনাৰ বেকগ্ৰাউণ্ডটো বৰ ধুনীয়া লাগিছে দিগন্ত দেই
এইয়া
চাল এটা
জিঅ'লজি মানুহ
জিঅ'লজি মানুহ ন তাকে ধুনীয়া লাগিছে
থেংক ইউ
হেৰি না মই আছো মই তাকে বোলো ভিডিঅ'টো অফ কৰি থওঁ বুলিহে কৈছো
ঠিক আছে ময়ো অফ কৰি লওঁ এতিয়া
ঠিক আছে থেংক ইউ
We will beginning the session shortly as our resource person has Dr. Ariel is not able to join the program due to some technical issues. So we will wait for a few minutes.
Hi, sorry, I'm a couple minutes late, but I just saw your email.
It's perfectly all right.
Sorry.
It's all right.
Okay,
We are just
perfect.
about to begin the session.
Okay, perfect.
Okay. Good evening, everyone, and warm greetings. I take the honor and pleasure to extend a warm and hearty welcome to everyone. Respected Principal Dr. Saibasasi Mahanta Sir, Vice Principal Dr. Reena Handik Pam, IQAC Coordinator Dr. Surajit Saikia Sir of Gorgaon College, respected Professor and HOD, Head of the Department, Dr. Diganta Bhuiya Sir, and Professor Kalpana Dekakolta Ma'am, Department of Applied Geology, Dibrugarh University. Today's resource persons, Dr. Ariel Pesnar, Postdoctoral Fellow at the Smithsonian Marine Station, US, and Dr. Utkarg Adhikari, Postdoctoral Fellow at Harvard University, US. We are honored to have such a distinguished group of faculties and participants joining us. I, Dr. Pakiza Begum, Assistant Professor in the Department of Chemistry, Gorgaon College, on behalf of the organizing committee, takes the pleasure to welcome you all to the 2 days online workshop, international workshop on career planning, exploring the recent trends in the ocean of science, which is jointly organized by the Department of Geology, Department of Chemistry, and IQAC Gorgaon College, Himoluguri, Assam, in collaboration with the Department of Applied Geology, Dibrugarh University, Assam, to mark the celebration of World Ocean Day. This 2-day workshop on 8th and 9th of June 2023 will include international talks from esteemed resource persons from prestigious universities of different countries. The main objective of the workshop is to provide a platform where students from different fields of science can explore career opportunities in reputed international organizations. This will help them in identifying their individual capabilities and systematically choose career goals to develop their future value in upcoming fields of research. To begin the workshop, I would now like to request eminent academician and principal of our college, Dr. Saibasasi Mahanta Sir, to present the welcome speech. Sir, please do the honor.
Thank you so much, Dr. Pakiza. A very good evening to you all. Dr. Diganta Bhuiya, Professor and Head of the Department of Applied Geology, Dibrugarh University. Dr. Chandraditya Gogoi, Head of the Department of Geology, Gorgaon College. Professor Kalpana Deka Kolita of Department of Applied Geology, Dibrugarh University. Dr. Anna Gogoi, Head of the Department of Chemistry, Gorgaon College. Dr. Reena Handik, Head of the Department of Zoology and Vice Principal of Gorgaon College. Dr. Surajit Saikia, IQAC, Coordinator IQAC of Gorgaon College, as well as today's resource person from different institutions. They have been joining us globally. Dr. Ariel Pesnar, Postdoctoral Fellow, Smithsonian Marine Station, USA. Dr. Utsarga Odhikari, Postdoctoral Fellow, Dana-Farber Cancer Institute, USA, Dr. Kolyan Raidongia, Associate Professor, IIT Guwahati, Dr. Sumit Dandapat, Postdoctoral Fellow, King's Abdullah University, Saudi Arabia, as well as esteemed participants of today's workshop, research scholars, and my dear students. I am delighted to welcome you all to this today's international workshop on career planning, exploring the recent trends in the ocean of science that has been organized by the Department of Geology, Department of Chemistry, and IQAC of Gorgaon College in collaboration with the Department of Applied Geology of Dibrugarh University. I would like to offer my sincere thanks and gratitude to the Department of Applied Geology for collaborating us to celebrate the occasion of the World Ocean Day and to organize this two-day international workshop on career planning, exploring the recent trends in the ocean of science, so that we can give a platform, we can provide an platform to our students as well as to our teachers and research scholars, so that they can interact with the eminent scholars, eminent researchers, and eminent experts in the field of study of oceans. As we all know, today is the World Ocean Day, and on this occasion, we would like to offer my best wishes to you all. And as we all know, that what is the importance of celebrating the World Ocean Day in the present day context, because we need our oceans for maintaining the balance of our environment, our ecosystem, as well as our sustainable future and sustainable environment. And as the oceans provide us the food, oxygen, and the oceans regulate the climate. And in the present day context, while we are fighting, we are confronting with the climate change and global warming, it is the need of the hour to protect our ocean, and this is the day to celebrate, to create awareness among all the need of the protection of oceans. And I would offer my I would like to congratulate the Department of Geology and Chemistry of Gorgaon College, who have been organizing this workshop in collaboration with the Department of Applied Geology of Dibrugarh University, to create an awareness as well as to provide a platform to our students, so that the motto of our college to developing human resource in this in the entire region, that college has been taken lots of initiative, and this is one of them. And as a part of our endeavor to create some students with create some researchers with some practical knowledge, this is a part of our endeavor, and I would like to congratulate the organizers for organizing such a beautiful event. And I hope the deliberations that would be taken in this 2 days workshop, the presentation that would be presented by the research scholars will definitely benefit our students, our teachers, and research scholars. I hope the 2 days this 2 days international seminar would be workshop would be a great success, and it will definitely pave a new way of thinking in the particular area of ocean of science. So with these few words, I hope the 2-day international workshop would be a great success. And again, I would like to welcome you all to this session, and I offer my sincere thanks and gratitude to the resource person for giving us some time from their valuable schedule to interact with us, to participate with us. With these few words, wishing you all the best. Hope the workshop would be a big success. And with these few words, I declare the session open, and I welcome you all to the 2 days international workshop on career planning, exploring the recent trends in the science of ocean. Thank you so much.
Thank you, sir, for your informative talk and encouraging us in organizing this program. Next, I would like to request the head of the Department of Applied Geology, Professor Diganta Bhuiya Sir, Dibrugarh University, to speak a few words on this important occasion. Sir, please do the honor. Sir, you have you have to put your mic on.
Okay. Good evening, everybody. The August gathering from the different parts of the world gathered today on this Zoom platform to celebrate World Oceans Day on this 8th of June 2023. Just after 3 days of World Environment Day. The oceans are very important to keep our planet safe and lively and save our environment in all the ways. And also the oceans are providing us all the economic assets, our foods, and our environment. They control our climate, all these things. So this day, the World Oceans Day, is very rightly celebrated just after the World Environment Day to keep our environment safe. So I'm very happy that the Gorgaon College of Himoluguri is organizing this World Oceans Day in collaboration with our Department of Applied Geology, Dibrugarh University, so that the students, the research scholars of this region, particularly the northeastern region, who has very less exposure to the science of the ocean, and they has very less knowledge about the different scopes of researches in ocean sciences. So I hope this 2 days workshop will open the avenues for the upcoming research scholars of this region to this very promising field of research, particularly this oceanography, then its allied subjects. Since our region is very much away from the oceans, very away from the seaside, so we have to carry out the researches in this new topic so that they are getting new scopes in this wide area of studies. With these few words, I wish this 2 days workshop will be a very successful one. Thank you.
Thank you, sir, for highlighting the importance of the occasion very nicely and giving us the opportunity for collaborating with your department. We are indeed privileged and honored to have amongst us today Honorable Dr. Ariel Pesner, who is a postdoctoral fellow at the Smithsonian Marine Station, US. She is currently studying the impacts of ocean deoxygenation on coral health. She has completed her PhD in biological oceanography from Scripps Institution of Oceanography at the University of California, San Diego. Her PhD research focused on characterizing the variability of water chemistry and dissolved oxygen and on coral reefs around the world, with particular interest of the impacts on coral calcification. She has a versatile research experience in different reputed laboratories of US, including the National Oceanic and Atmospheric Administration, where she achieved the prestigious PIFSC Young Scientist Opportunity During her PhD, she was NSF GRFP Fellow in the Scripps Coastal and Open Ocean Biochemistry Lab with Dr. Andreas Anderson. She has tremendous research contributions on emerging fields like temporal variability in seawater carbonate chemistry, biochemical variability with coral reefs, and climate influence on different marine phytoplankton, including a recent publication in the reputed journal Nature Climate Change. And before I hand over the session to the esteemed speaker, I would like to make an important announcement that filling the feedback form for all the sessions and attempting the quiz at the end of this program are necessary to generate the e-certificates. Also, queries are most welcome and you can type them in the chat box as it will facilitate us for a smooth conduction of the program. Now, let's open our mind and heart to new ideas as we come together to learn new facts and share knowledge. With this, I hand over the session to Dr. Pesner and like to request her to give her speech.
Thank you so much for that introduction. Yeah, I'm honored to be here today. Sorry again that I was a couple minutes late. I have a PowerPoint presentation, if I can share that.
Yes, you can, I think. We have to make our co-host. Yes, you can share now.
Okay, perfect. Okay, yeah, so I thought I would just give a little bit of an overview talk of my kind of subfield within oceanography, which is coral reef biogeochemistry. So kind of looking at the different applications and opportunities. It sounds like today is a good opportunity for a lot of you to kind of see what different options you have when thinking about ocean science. And so, yeah, I wanted to hopefully kind of show you a little bit more about my field and how maybe you can get involved with it if you're interested. But I wanted to start with a little bit of my own personal background and kind of how I got to where I am today. So I grew up in Los Angeles in California, and I attended UCLA for my undergraduate degree in environmental science. And while I was there, I actually ended up minoring in atmospheric and oceanic sciences, as well as conservation biology. And I also got involved in research as an undergraduate, which was really important for my career and kind of exposed me to a lot of different fields so that I could kind of get a sense of what I was most interested in. So I actually started on land, not in the ocean. I worked in a terrestrial biogeochemistry lab. And so at this lab, we were looking at the impacts of California's drought on the photosynthetic capabilities of native plant species. And interestingly enough, this instrument that I'm using here in the field to measure photosynthesis on land plants, I'm actually using during my postdoctoral fellowship here to look at photosynthesis in corals. So even if you think the two fields may not be totally connected, you'll never know what instruments kind of carry over between like land and ocean spaces. While I was an undergraduate, they also had a field research program. And so I got to travel to French Polynesia to do a couple different field projects. I looked at the impacts of different stressors on marine macroalgae. So this is a picture of all our different algae in these different treatment cups that were set in water tables. And I also looked at the hiding behavior of Christmas tree worms, which are these little marine worms here that burrow into coral heads. And so that was more of like an animal behavior project. So I tried out a lot of different fields of research kind of before I settled on coral reef biogeochemistry. I also did internships, like was mentioned in my introduction. I worked at the NOAA office, so the National Oceanic and Atmospheric Administration. I did more of a physical oceanography internship one summer, looking at the movements of ocean fronts. And then I also did one more focused on plankton dynamics in Narragansett Bay in Rhode Island. So all of these different experiences kind of showed me what aspects of science I liked and what aspects I didn't like. And so all of those experiences were really valuable in kind of shaping how I thought about research and what I was interested in. And so I was lucky to end up attending Scripps Institution of Oceanography, which is part of University of California, San Diego, so down in Southern California. And I really liked field field research and being able to do field work. And so a lot of my PhD, all of the data that I collected kind of came from field work that I was able to do in the first couple of years. So we were kayaking over reefs, diving on reefs, putting out instruments on reefs, and doing a lot of work on reefs, which I thought was really special. And then we kind of bring all of that data back to the lab. And my PhD technically is in oceanography, but really I was looking at reef biogeochemistry, which I'll talk a little bit more about like what that really means. And then now I am a Smithsonian postdoctoral research fellow. So the Smithsonian is a really big kind of museum complex. So if you've ever been to Washington, D.C., they have a lot of natural history museums there. But Smithsonian also has a marine station down here. I'm in Florida in the USA, and I'm working on some more kind of biologically focused experiments with corals. This is a picture of me and a couple of my interns preparing corals for an experiment and measuring and taking different photos of them during the course of the experiment. So it's a lot of different kind of fields coming together to make my career. So just like a little bit of background of why we study coral reefs. They're globally important marine ecosystems. They cover less than 2.2% of the seafloor, but they're home to more than 25% of all marine species that we can find in the ocean. More than 500 million people around the world depend on coral reefs for a source of food and income, and they also protect coastlines from storms. They provide over $2.7 trillion in goods and services and $36 billion in tourism money every year. And they're also really important for absorbing wave energy and preventing flood damages on coastlines. But as I'm sure you know, hard coral cover is declining worldwide. So this is a plot from a recent Status of the Corals of the World report, where you can see this kind of variability in coral cover around the world, but this kind of general decline, especially in the last decade. And this is due to a variety of different reasons. There's coral diseases, which a lot of people at the station here are studying. Overfishing, so removing of fish leads to less healthy coral reefs. There's also pollution from land and runoff, as well as just physical damage from people trawling or destroying reefs. But what I kind of more focus on and what I'm more interested in is how global climate change presents a significant challenge for coral reef survival. So this is the famous Keeling curve, which just shows this increase in CO2 concentration over time. And it varies seasonally, but in general, we have this general increase of CO2 due to our release of fossil fuels into the environment. And this has several different consequences for ocean environments. One, of course, being global warming, which we see reflected in the sea surface temperature, where we see this increase in sea surface temperature over time. And these different colored lines just represent potential projections for future change. So we could see, in the worst possible scenario, up to a 4 or 5 degree change in seawater temperature by the end of the century. But still, regardless of, even if we cut off all of our CO2 emissions right now, we would still see a pretty significant change in sea surface temperature. We also see this decrease in ocean pH, which is called ocean acidification. This is kind of what I initially came into graduate school looking really interested in studying. But there's a lot of people studying this kind of field of ocean acidification, and it's a pretty saturated field. But kind of a less studied field, and what I ended up kind of focusing on and what I've kind of carved out as my niche within science is looking at ocean deoxygenation. So I think a lot of people realize that there's this increase in warming and this decrease in pH, but we're also seeing this decrease in ocean oxygen content. And so this is a side effect of this warming that we see, which I'll talk about a little bit more later. But in general, we're seeing this oceans that are increasingly warm, acidic, and low in oxygen. And so, as you know, the warming can cause bleaching and mortality. Warming is what causes what we can see like on reefs, where we go from these really colorful, vibrant reefs to these kind of stark white reefs, which is that process of coral bleaching, which is mostly driven by temperature. And ocean acidification can pose a threat to corals as they are marine calcifiers. So this addition of CO2 into the seawater leads to this series of dissolution equations. But overall, if we're adding more CO2 to the ocean, there's less carbonate ions available for corals and other mollusks and things that calcify and make these hard skeletons. They're less able to do so. And so this also decreases the pH of the ocean and makes it more acidic. And so, as I mentioned, warming is also leading to global ocean deoxygenation, which is kind of my more specific field of study. So typically the ocean gets oxygen by this mixing with the atmosphere and equilibration with the atmosphere. However, when we have a warmer surface ocean due to global warming, this leads to a decrease in the solubility of oxygen in water, just because like warmer water can't hold as much gas. We also see this level of stratification, which means you kind of have this lens of warm water sitting over the cooler water, and it prevents this mixing with the atmosphere. And we also see an increase in respiration, just because respiration tends to increase under higher temperatures. And so overall, in the last 50 years, we've seen that the ocean has lost about 2% of its oxygen. And this is especially true, as you can see, in tropical areas where we can find coral reefs. And so kind of the two main chemical species that I'm interested in, although there's definitely many fields of chemical oceanography that study a whole range of other chemicals, but mostly we're looking at oxygen and CO2. And so when we think of a coral reef, right, there's corals living there which photosynthesize, there's marine algae that photosynthesize, and then there's all the other animals and stuff that live there that are respiring. And so during the daytime, when we have sunlight, oxygen is being released into the water through photosynthesis, and CO2 is being taken up. But then we also have this ongoing process of respiration where oxygen is drawn down and CO2 is released. And these usually lead to this kind of diel cycle, so this daily cycle that we would expect to see on a coral reef, where during the daytime, oxygen is high and pH is high. And then at night, with this oxygen level goes down and the pH goes down, and this kind of repeats generally every day. But the shape of this curve and the amplitude of it and a lot of other things about it are dependent on what is on the reef. And there's also other things that can influence the pH, for example, dissolution and calcification. So if these corals are making their skeletons or if they're dissolving, it will also affect kind of the trend in pH that we see on a reef. And so by measuring something such as oxygen or pH over time or across the reef, we can kind of get a sense of like what is happening on the reef and which of these processes are kind of dominating what's happening. And so this is kind of like applications of coral reef biogeochemistry. So one of the main tools that we use to kind of capture this variability is autonomous sensors. So these are just basically instruments that we can put on the bottom of the reef and kind of attach there, and we put it in one place, and you can leave it like up for a year, maybe 2 years at a time. And this allows us to capture high frequency variability just in one place over time. And so, for example, a case study from one of my dissertation chapters was looking at this place called Dongsha Atoll, which is in Taiwan. And we focused on the seagrass bed that was on the north shore of the island over here, which is this really cool environment that had seagrass and also corals growing together in it, which is kind of a unique place. And there were corals growing amongst this like really dense seagrass bed. And we kind of expected there to be interesting chemistry going on here, just because with all this seagrass, we'd expect a lot of photosynthesis going on during the daytime, but also a lot of respiration happening at night. And so we ended up placing an instrument in the seagrass there and left it there just for a week. And from that data, you can see this kind of diel change that I was talking about. So for example, we can see that during the daytime, the temperature is high because the sun is warming the ocean. The dissolved oxygen is extremely high, and the pH is also quite high, above 8.2. But then we see at night that the oxygen goes down quite significantly, and it's actually severely hypoxic every single night that we had our instrument out there, which is this kind of red bar here. And we see that there's pretty significant acidification happening every night, where the pH is changing more than 0.4 units every single night, which is a really big change. And, in the open ocean, if you were to record something like this, it would be a much smaller amplitude and change from day to night. So these corals are experiencing really big swings in both oxygen and pH overnight. And so these instruments can tell us something like that, where you have one spot and you look at it over a really long period of time, and you can see, what that reef is experiencing, on day to day or an hourly or even minute basis. But then it kind of begs the question of whether this variability is maintained over space and across time, because if we just put the sensor right here, does that sensor really tell us about what's happening in this part of the seagrass bed or over here or even outside of the seagrass bed? And so to kind of get that spatial aspect of this variability, we also tend to perform spatial surveys, which help us capture the variability over space and time. So for example, on the seagrass bed, this purple diamond is where we deployed that instrument and got that time series. But then we also complemented it with spatial surveys. So we collected water samples at these different gridded locations over the seagrass bed and outside of the seagrass bed, both in the early morning and in the late afternoon and then the mid-afternoon, to kind of capture what this looks like over space over different times of day. And so, for example, we can see strong gradients in the oxygen across the seagrass bed. So these plots are showing an early morning survey and a late afternoon survey, where low dissolved oxygen is in this blue color. And you can see in the morning, oxygen is extremely low across most of the seagrass bed and even severely hypoxic in the shallowest parts. And then in the late afternoon, the oxygen gets up really high, especially in the shallowest parts. And so you can kind of see that even though this matches like the data that we got from our sensor that was deployed at the shallowest site, you can see that there's a different structure and a gradient that goes from inshore to offshore and can tell us more about what corals in different parts of these seagrass are experiencing. And so we can do that. We can analyze these water samples and look at changes of pH across the seagrass bed, and also some other aspects of carbonate chemistry like dissolved inorganic carbon and total alkalinity, which can tell us more about if photosynthesis is happening, and similarly, TA can tell us a little bit more about calcification and dissolution. And so this is all kind of matching our like observations of the biology of what's actually happening. You know, if you were to stick your head in the seagrass bed, this is what you would see. And you can see that it's extremely productive. You know, you can see the oxygen bubbles kind of like bubbling off of the seagrass during the daytime. And so overall, understanding present day variability of both CO2 and oxygen on these reefs is really needed to make future projections. And these are all complicated by different factors. So who is there on the reef, the benthic community, you know, if you have a lot of seagrass, you're going to see a lot more variability than you might on a reef that doesn't have as many primary producers. It's also affected by the geomorphology on the reef. So for any geologists out there, you know, thinking about the shape of the reef, if it's really shallow, if it has a sill that prevents it from the water being exchanged with the open ocean, and also the type of rock that's there kind of influences how the CO2 and oxygen varies on a reef. And physics also plays a really strong role in a lot of the work that we do, kind of thinking about hydrodynamics. So how long is the water sitting over the reef? What direction is the flow going? All kind of impacts these measurements that we make. And so ocean biogeochemistry is really an integration of both the biology, the geology, the chemistry, and also to an extent, the physics of the reef and how that affects the patterns that we see. And then this is, of course, all complicated when we add on the effects of climate change. So how does changing temperature affect all of these different factors and the variability that we see now and also would project to see into the future? So kind of an example of this like application of autonomous sensor data comes from this paper that I recently published in Nature Climate Change. So I just wanted to give a quick summary of it just so you can see how we can use this autonomous sensor data to make projections about how conditions will look in the future. So typically coral reefs are assumed to be well oxygenated systems. You know, they have these like crystal clear waters and they have lots of primary producers. And so we expect the oxygen to be high. But as you saw from that sensor data in Dongsha, this oxygen might be high during the daytime, but it's also could be extremely low at night. And there are also severe hypoxic events that happen on coral reefs that lead to mass migration of reef invertebrates to the surface, as well as bleaching and mortality of the corals themselves. And so these hypoxic events are predicted to be increasing across coral reefs, but there's not a lot of evidence to show like what oxygen looks like on coral reefs today. And so for this project, we utilized this global coral reef data set that involved data from 33 different reef sites at 12 locations around the world. Of course, it doesn't actually represent all of the places in the world where we have coral reefs, but these were the data that we could get at the time. And so these data came from a variety of different coral habitats. We had patch reefs, we had seagrass beds, and we also had data from deeper reefs and also shallower reefs that were all recorded by the same types of sensors, like the sensor that I showed before. And so using these data sets, we could then leverage location-specific temperature projection data from global climate models, which is what CMIP-6 is. It's a global climate model that lots of people use. And for each location, we extracted what the different temperature projections would be, and then we calculated what the decrease in oxygen solubility would look like under these different temperatures, and also the increase in reef respiration rates under these different temperatures. And overall, we could show that the availability of oxygen is going to decrease on these reefs under warming. So this plot shows the mean range in dissolved oxygen in blue at present day, and under these different increasingly warm scenarios to red for each of our different reefs. And so, for example, if we look at this reef in Bermuda, which has quite high oxygen levels today, we'll see that under warming and even the most severe warming scenario, like a 6-degree heat wave, we would see that the oxygen availability would go down. However, it would still not be considered hypoxic as we define it today. But if we look at Dongsha, the reef that I mentioned before, we can see that even at present day, Dongsha is experiencing low oxygen levels at night in the seagrass bed, and that under these increasingly warm scenarios, this reef would likely experience low oxygen levels almost all of the time. And so overall, this shows that more reefs than we would have expected. So almost 84% of these reefs that we surveyed experience weak to moderate hypoxia or low oxygen conditions under present day. And just about 13% of them experience severe hypoxia, but this would increase to almost all of these reefs and about a third of them experiencing severe hypoxia under the warmest climate scenario. And so this kind of jumps into the more biology focused part of my research is kind of, we can show what's happening on these reefs and we can predict what might happen to the ocean water surrounding these reefs, but what does it really mean for the corals themselves? Well, unfortunately, the thresholds of hypoxia, so kind of like what levels of oxygen matter to different reef organisms, is not really well resolved. This is a figure from a recent paper, and I added in a couple more citations from recent papers, but really this is all the data we have on what are the important dissolved oxygen values for a bunch of different species. So there's a lot of data on fish. There's a few data on corals, but you can see that some corals are very tolerant of low oxygen, whereas some corals are very intolerant, and we don't have a lot of data for any other reef organisms. And so understanding which level of oxygen is going to be relevant to different organisms is going to be really important for predicting how the reef community or the biology of the reef is going to change under low oxygen. And so there's also this difference between lethal levels. So, the point at which these organisms just die versus sublethal levels. So the point at which maybe they stop growing or they're not respiring as much or they change their behavior. And we also don't have a lot of information on, how long do they need to be exposed to these oxygen levels to see these effects, or how often they need to be exposed. And so in addition, of course, as I mentioned before, this warming and hypoxia are not occurring in isolation. We also have this background effect of ocean acidification, and acidification and hypoxia actually really tend to happen in the same time and the same place. So a lot of the factors that lead to these like severe hypoxic events on reefs also lead to low pH values because there's a lot of respiration happening. And data has shown that warming actually increases organism sensitivity to low oxygen. So under higher temperatures, they get more and more sensitive to low oxygen values. And so really we have to think about all of this in kind of like a multi-stressor context. And so that's what the focus of my postdoctoral fellowship is about, is looking at the effects of all three of these stressors together on corals and coral biology. And so some more ongoing work that I'm working on is looking at using seagrasses as natural laboratories, so the seagrasses on Dongsha, and also looking at genomic changes of corals under multiple stressors. So looking at these kind of effects of highly variable oxygen and pH regimes on coral growth, and also the physiological and genomic effects of hypoxia and low pH exposure. And so, for example, in the Dongsha seagrass, we drilled coral cores from quite a few of these corals. And this is kind of the more geology part of the biogeochemistry, that you can take these cores from these corals and X-ray them. And from that, you can extract growth rate data. And then we also extracted stable isotope data from these corals to see how their use of carbon has changed over time, and also what the temperature was like in the water when the coral was growing. And then I'm using Aquaria set up here to look at the physiological and genomic effects of hypoxia and low pH exposure on corals, as well as temperature. So this was kind of just like a general overview of biogeochemistry. So there's definitely the applications of biology to look at how these different stressors affect corals, or really any other reef organism on a biological or molecular level. There's the kind of geology aspect of the shape of the reef, the hydrodynamics, as well as looking at the corals themselves as they kind of create these calcium carbonate skeletons. And then, of course, a lot of chemistry applications for inorganic chemistry and looking at carbonate chemistry on reefs. So Yeah, thanks for listening. I'm happy to take any questions you have, or if you have any questions about the science or even about my path in science, I'm happy to answer them.
Yes, we have got one question. This is how is BioRock compared with conventional BioReef compared with conventional artificial reef, and how is it formed? This is asking about BioRock reef in comparison to conventional artificial reef.
Yeah, that's a great question. I think there's quite a few different groups working on developing those. It's not my specialty, but from what I understand, it seems like a conventional artificial reef can be made out of anything, right? Like it could be, in Florida, they had a reef where they just threw a bunch of tires in the ocean and hoped that it would create an artificial reef, which I wouldn't recommend. But other people, use concrete or other kinds of structures, or even like metal structures can be artificial reefs. But it sounds like BioRock-based reefs, are specifically made to be made of the same things that corals are made out of, or perhaps have like the algae or some sort of like probiotics or something like that, like injected into them to make them more like likely for corals to settle on them. Yeah, it's not my specialty, but that was like my understanding of what a BioRock reef is.
Currently, we don't have other questions. Your talk is very interesting. I think this is the first time we have got the exposure to see such kind of work, how the climate change, the global warming is affecting these coral reefs. This is very interesting, and I'm sure the students will be having more questions later on when they, actually we have got one more question. Can I? Does coral gardening help to mitigate the current scenario?
Yeah, that's a great question. It's something I've been thinking about a lot. I think before I moved to Florida, I maybe was not as convinced that outplanting was the solution to saving coral reefs. Because, in my mind, if we're not fixing the environment and we're just putting more corals back out there, then they're maybe not going to be, ready to handle it. And if we're just, not changing our activities and letting the ocean continue to warm and continue to be low oxygen and low pH, then those corals are, maybe not going to survive. But, in places like Florida and the Caribbean, where these coral diseases are really wiping out a lot of these corals, outplanting corals that are more resistant to them, maybe a good solution. So I think it's kind of like one part of the bigger toolkit that we need to use to address these problems on coral reefs.
Thank you. We have another question. How do ocean acidification and warming affect the health and survival of coral reefs?
There's a lot of literature on those two stressors. I don't think I can really summarize it for you here. I mean, in general, I think, warming is a really immediate, severe threat to reefs. Like, coral bleaching is a really big problem. It happens very quickly. And it's like marine heat waves, for example, on reefs are very severe. And, bleaching is very bad for corals. It's very hard for them to come back after that. Ocean acidification, I think, is maybe not as immediate of a threat, but I think over the long term will make it increasingly hard for corals to grow their skeletons.
And what about Coral Reef Alliance? One more question.
Yeah, I'm not super familiar with their work. I believe that they're like a nonprofit kind of working to kind of synthesize coral reef conservation activities. And, I think all the more people that care about coral reefs is for the better. And like, maybe uniting kind of all of our efforts together to common goals is going to be the way that we conserve coral reefs.
Another question. I think the students are very interested.
Yeah, of course.
How do coral reefs absorb wave energy? Is it related anyways to tectons or mitigation to tsunami?
Yeah, coral reefs really absorb the wave energy just by being like a physical barrier. You can look at, there's probably some like videos online that kind of simulate it, but basically if you have corals there, they have this like really complex structure, they're kind of like shaped like trees or like fingers and stuff, and they create kind of like a physical wall, so that when the waves hit these corals and they break over these corals, they lose a lot of their energy before they make it all the way to the shore. And so if you remove the corals from being there, then there's nowhere for these waves to break except for on the beaches or on people's houses that are right along the coast, or whatever is there. And so they absorb a lot of the wave energy. I think when it comes to a tsunami, I'm sure they help a little bit, but probably not, not enough to mitigate a tsunami completely. But I'm sure having it there is better than not having it there.
Another one. Does mineral accretion improve coral reefs?
I mean, corals need to calcify and grow their skeletons and able to survive and to keep growing. So yeah, the accretion of calcium carbonate within the corals themselves is really important, which is why people think that ocean acidification is a problem, because it kind of reduces the amount of carbonate that's available for corals and other, calcifying organisms to make their skeletons.
Another one is there any lifespan for the typical biorock reefs?
I don't know personally. I assume that, as long as they're out there and things are growing on them, then they could last a long time. But yeah, unfortunately not my specialty.
All right. Another one, what will happen if all the coral reefs wiped out from the oceans?
Probably not many good things. I mean, we rely on them for, as I mentioned before, like a lot of people depend on food that comes from coral reefs. It's a source of a lot of money for tourism for people in like Australia or, in the Caribbean or, anywhere that has coral reefs. So yeah, I don't like to think about it. Definitely not good things. So I think we should work hard to conserve them so that doesn't happen.
Yes, our students have gotten exposure to such fields through your presentation, and this definitely opens up more career scopes that they can explore in their future. We are indeed very much thankful to you for your nice and informative presentation.
Of course. Yeah, thank you for having me.
Thank you. Now we have with us our second speaker, and we are privileged to have him with us, Dr. Utkarg Adhikari, who has joined us from Dana-Farber Cancer Institute in Boston, USA. We extend a hearty welcome to you, sir. Before handing over the session, I would like to give a brief introduction of him. Dr. Adhikari is a postdoctoral research fellow in the Walensky Laboratory at the Dana-Farber Cancer Institute in Boston, where he studies key protein-protein interactions in the critical physiological process, cell death, and the cell cycle in the context of cancer. Sir is passionate about cancer research as well as mentoring the next generation of science graduates, and has served as a mentor for numerous summer students and interns, both interns. Additionally, he has taken a keen interest in professional development for graduate students and postdoctoral scholars, and is an co-creator and co-developer of the Graduate Bioscience Career Cognitive City at Harvard Medical School. This is an interactive website that organizes hundreds of career resources for academic trainees. Dr. Adhikari received a BSc in chemistry from the University of Calcutta, India, MSc in chemistry from Indian Institute of Technology, Guwahati, and a PhD in biological and biomedical sciences from Harvard University, Boston, USA. Sir, I would now hand over the session to you.
Thank you very much for having me. Can you hear me properly?
Yes, you are audible.
Okay. Okay. I'll try to share my screen. Okay, is that visible?
Yes, it is visible.
Okay. Okay, great. Yeah, thank you, first of all, to all the organizers for having me as a speaker. I'm excited to share my research that I did during my PhD. Thank you also for the introduction. As you or I actually did my master's in Assam, in IIT Guwahati. So it's like nice to share some research for my PhD after coming to the United States almost 7 years ago. So I'll talk a little bit about research, and since this is a workshop on career planning, I'll cover some pointers on of career planning and some lessons I've learned along the way, which you might find helpful as undergrads. So in the lab, in the Walensky lab where I'm a postdoc, we study the essential process of cell death, which is the central question of like, how do cells in the human body die? And we do that in the context of human cancer. As you can imagine, in cancer, what happens is that there's a lot more cell division that occurs and not enough cell death, which is how tumors form. So I'll try to give an overview and try to explain my research in simple terms, because I understand the audience is not a biology audience. So I'll try to make sure to explain it in a more lower level. But so as an introduction, you know, how does the cell die? So like in the cell, when it comes to the process of cell death, in the nucleus, the cell condenses its chromatin, which contains all the DNA. And this is followed by a process known as membrane blebbing. And then it forms what we call apoptotic bodies, and the cell basically disintegrates and dies. To make it a little bit more clear, I have this little video here. And so you can see with the red arrows, these are like two dying cells. So like they condense, they have these bubble-like structures, so that's the membrane blebs, and they finally disintegrate and die. And so in the lab, we're really concerned about this process. And as you can imagine, it kind of regulates a critical balance between life and death. So on the left, like when your cells, the number of cells that are forming and the number of cells that are dying are in balance, you have a homeostatic condition. This is essential to maintain because, as you can imagine, if you have too many new cells forming but not enough cell death, you can have diseases like cancer. And then similarly, if you have too much cell death and not enough new cells replacing that, you can have diseases like neurodegeneration or immunodeficiency. And so there's a group of proteins in the human body that regulate the function of cell death or apoptosis. And this group of proteins is called the BCL2 family of proteins. And within the BCL2 family of proteins, you know, it's subdivided into three main classes of proteins. So in this slide, depicted in red, are what we call the anti-apoptotic proteins. So apoptosis means cell death. Anti-apoptotic refers to proteins that help a cell live. So these are important proteins for a cancer cell because a cancer cell wants to express more of these proteins because they want to live. They don't want to die. So they do this by increasing the levels of these proteins in the cell. But to keep these proteins in check in a normal human cell, you also have these class of proteins in gray, which are the pro-apoptotic proteins. And their job is to keep these red guys in check and control so that, you know, the cells don't divide without any control. And then there are the BH3 only proteins, or smaller proteins that are depicted in blue here, that can either oppose the red anti-apoptotics or engage the pro-apoptotic proteins. So together, these proteins kind of regulate the process of cell death. And this background is important because the protein that I studied for my PhD is called MCL1, which is an anti-apoptotic protein. So it promotes cell survival in cancer. To give you a little bit of context of how this happens in the cell, This on the left is a diagram of a cell, and within the cell there are different organelles. Like there's the nucleus, there's cytoplasm, and most of the process in cell death is initiated in the mitochondria, which is depicted in pink. And then within within the mitochondria where cell death takes place, there's also this protein called MCL1 that I just talked about in like 2 slides ago, which is an anti-apoptotic protein. And as it turns out, research has shown that MCL1 is one of the top 10 most commonly amplified or overexpressed genes across a broad range of human cancers. So if you look at lots of human cancers, different types, and you look at what proteins do these cancer cells upregulate the most, you will find MCL1 in the top 10. So that makes it a very critical target in cancer, and therefore it makes it important to develop compounds, chemical compounds, small molecules, and treatments against this protein. So the way that this works on the mitochondria, as I mentioned, is that you have this mitochondria, and MCL1 here depicted in these red blobs sits on the outer mitochondrial membrane, and it can oppose the function of BAX, which is a proapoptotic protein, if you remember from that slide in gray. So the red proteins block the gray proteins and prevent them from carrying out their death function. And this is how overexpressing of this protein creates a block on the cell death program in cancer. And what happens due to this is that it leads to development and resistance in cancer. But the interesting thing about MCL1 is, in addition to its role in cell death, over the last several years, we found that this protein has many other functions within the cell. And these could be mitochondrial energetics, fatty acid oxidation, hematopoiesis, which is the formation of blood cells in the body, or cardiomyocyte contractility. So, when your heart is beating, the heart is made of cells known as cardiomyocytes. And MCL1 has a role in the contractility of these cells as well. Overall, this just goes to show that MCL1 is emerging as a very critical protein in normal physiology. And so to give you an overview or a context of like how we did the research on MCL1, so basically we want to study the function of MCL1. I did my PhD mostly focused on the cell cycle role, and I did this primarily in 2 ways. So one way is we can do genetic deletion experiments. So when you want to figure out what is the importance of a protein or a gene in the cell, like why is it important? One way that biologists like to ask or address this question is, let's delete the gene or take away the protein, and then we'll see what happens as an effect of that. So, if you delete the MCL1 gene, it allows you to study the effect of the total absence of MCL1. So when you take away something important, something bad's going to happen, and you can study that and figure out the mechanism. The other way to do that is if you have a small molecule, like a chemical compound that you synthesize, and you can block the function of this protein, you can use this compound and you can block the function of the protein. And we have a molecule that can bind to the binding groove in the MCL1 protein. And so that allows us to study the effect of pharmacologic targeting of MCL1. So gene deletion and small molecule inhibition are 2 ways we can do that. So in my research, we started out by studying the effect of gene deletion that I just mentioned. So you take away MCL1 from a cell that is dividing, and you see, and what we saw was that the cells that are deleted from MCL1 can't grow as fast. They don't proliferate in culture. So in black is like wild type cells or normal cells. And in red is like cells where you take away MCL1. And as you can see with time, there is less of the red cells that are growing. So this protein seems to be important in cell division. And we showed that with a different knockout system. So on the left is a more permanent knockout where you delete the gene permanently, and in the middle panel now is like you can delete MCL1 temporarily. And you see the same thing where you delete it temporarily, you get a decrease in cell proliferation. And then we can put the MCL1 back into the deleted cells. So you essentially take something away and then put it back in, and you're able to restore the phenotype. So that validates the function of MCL1 in cell proliferation. And we also found that, so there are two different types of this protein, or isoforms. One of them is on the outer mitochondrial membrane, so the outside membrane of the mitochondria. The other one, other form of the protein stays within the mitochondrial matrix, or the inner parts. And we found that when we took these knockout cells in red and put back only the part that resides in the matrix, or the inside, it wasn't able to restore the cell division. But when we took the outer mitochondrial membrane form and put it back into the cell, then it was able to partially rescue. So this indicates that it's the part of the protein that resides in the outer mitochondrial membrane that's responsible for this function. So that was all doing genetics, deleting the gene. As I was mentioning, the other way we do it is take a small molecule. So we have a small molecule that we've used for the studies. It's called S63845. And this small molecule can bind the binding pocket of MCL1 on the protein surface, and therefore block any interactions of MCL1 with other proteins. So the central question we had after seeing an effect of genetic deletion is, does the same thing happen when you add a small molecule? So does small molecule inhibition phenocopy genetic deletion? And the answer is yes. So in different cell lines that we tested, when we treat these cell lines with increasing doses of the MCL1 inhibitor S63845, so it goes from gray, which is no treatment, and then different increasing shades of blue corresponding to higher doses of treatment. And as you can see, the higher you go, the more the cells are the less they are able to proliferate. So this indicates that blocking the protein is similar to taking the protein out by genetic deletion. We also wanted to ask how deleting this protein in a genetic fashion in an organ leads to what kind of effects does that have? So we analyzed an RNA-seq data set that compared thymus, which is an organ in the body, in mice. So these mice were either normal wild-type mice or mice where MCL1 was deleted. And we took the thymuses and we asked, which genes are upregulated or increased in the MCL1 knockout case, or which are decreased in the MCL1 knockout case. So like what are the genes that are differentially expressed when you do a knockout in the thymus? And then using those genes, you can kind of infer what pathways in the cell are upregulated or downregulated. So what we found was a number of cell cycle genes, or genes that are very important for a cell's division, were upregulated when you took this protein away. So in MCL1 knockout thymuses, we found that there was an upregulation in ribosomal biogenesis, cell cycle, and DNA replication pathways. And some of these examples are shown in green here. And so as to stick to the theme of testing genetic deletion and then small molecule inhibition, we then switched our efforts towards figuring out the effect of inhibiting MCL1, but in a much broader context. So in Boston, there's an institute called the Broad Institute, and they have a very big parallel platform to test different chemical compounds against different cancer cell lines. So we submitted the MCL1 inhibitor in this assay, and it was so they basically screen your molecule against 1,000 cancer cell lines, and so that enables you to then not only see what kind of cancer cell lines are susceptible to your small molecule, but also how your compound compares to all the other compounds that they have in their data set. So in their data set, they have almost 500 small molecules, chemical compounds that target different proteins in the cell for different functions. And so if you, let's say you went to the chemistry lab, you made a compound and you submitted it to this assay, it can tell you how does your compound compare with other compounds in the data set, which pathways does it hit, et cetera. So what we found here was that the, the question we were trying to ask is like, what is the pharmacologic profile of the MCL1 inhibitor across the cancer cell line encyclopedia and how it compares to other small molecules? And we found that, on the left, which is the S63845 MCL1 inhibitor, it's most similar, its profile is most similar to compounds that target the cell cycle or anti-proliferative drugs, which are colored in blue. In contrast, if you took a small molecule that targeted another anti-apoptotic protein in the same family, so like MCL1's brother or sister, so it's like a similar function protein, if you took that compound, you found that it corresponded more towards standard apoptosis drugs, which are colored in red, which is what you would otherwise expect. But we found that the MCL1 inhibitor, interestingly, hit or looks more like a cell cycle drug than an anti-apoptotic drug. And then when we compared the gene expression that I showed a couple of slides ago in MCL1 knockout mice versus wild type, it resembled treatment with the microtubule targeting agents, vincristine, oxybendazole. So in essence, this meant that when you knock out the MCL1 gene, it resulted in these gene expression changes that resembled as if you treated with a microtubule targeting agent. And so microtubules, for those of you who don't know, are essential structures within the cell that help the cell to divide. And so when you disrupt the microtubules, cells can't divide as well, and ultimately they die. And so having found this similarity of MCL1 deletion and microtubule targeting agents, we conducted a screen in the lab drug screen where we took a number of FDA-approved cancer drugs and took wild type or MCL1 knockout cells and treated them. And we found that microtubule targeting agents, phenylalanine, vincristine, and paclitaxel in blue here, killed the MCL1 knockout cells much, much better than a wild type cell. So you can see, panels B, C, and D, where we validate that. Like, wild type cells, the cells are more alive, but in MCL1 knockout cells, these are like really dead. And then when we took MCL1 and put it back into the knockout cells, so in green in EFG, you can see that the cell viability goes back up. So you took away a protein and it caused the cell to die more, and you put it back in and it restored its viability. So that told us that MCL1 deleted cells have a special vulnerability or susceptibility to microtubule targeting agents. And we validated this with combination treatment of S63845 and vincristine. And in the co-treatment cases, there's much more cell death that we see. And this corresponds to also an increase in cell cycle arrest. So you can do some experiments where you can figure out how much of your cells are arrested while trying to divide. So when there's a problem, cells enter what is known as cell cycle arrest. So they pause instead of completing the cycle. So we saw that in case of the combination in deep red here, there's more cells that are arrested in the G2M phase. And we expanded our study to different cell lines and different MCL1 inhibitors and different microtubule drugs, and did all of the possible combinations to see if what we found was indeed general across different cell lines and across different classes of drugs. And in each case, we found that the combinations proved to be more lethal to the cells. So the more red it is, the more the cells die. So in all cases, the combinations prove better than the single agents alone, which shows that targeting MCL1 and the microtubule is a viable combination strategy. So that's the positive side of when you think about cancer therapy. You want to find combinations that kill a cell more. But we also have to think about the flip side, which is that, how do we make sure, like, we want to make sure that combinations are not toxic. So like they won't kill normal cells. So to do that, we have begun some preliminary toxicology experiments in mice where we treat the mice with either vincristine or S63845 alone, or the combination. And we found that actually in the co-treatment cases, so in purple here, the mice that receive the co-treatment suffer from more significant weight loss than the single agents, which could indicate that the co-treatment definitely causes some sort of toxicity that we should be aware of. And this is important because the S638 molecule is now in phase 1 clinical trials. So it's important for the community to understand the risks involved in addition to the benefits, obviously. And then because cell cycle is intertwined with DNA damage, we also took a look at how inhibiting MCL1 can cause DNA damage. And we saw that when we treated cells, so this is a Western blot, which is a common technique used in biology where you can detect the levels of proteins or expression of proteins in a cell lysate. So we saw that in wild type or normal cells, when we increased the dose of S63845, we saw more signal of gamma H2AX, which is a protein that marks DNA damage. So the more gamma H2AX there is, the more there's DNA damage. And we saw that in a wild type cell, when we knock out the cell, knock out MCL1 in the cell, so you take the target away, you no longer see that effect, which is good because that means the effect you see is on target. Without the target, you don't see the effect. And then when you reconstitute MCL1 back into the knockout, you again restore that trend of gamma H2AX induction. And finally, we took leukemia cells or blood cancer cells that were either wild type for MCL1, labeled as DKO in blue here, or deleted for MCL1, labeled as TKO in red here. And we found that when we put these cells into a mouse, the cells that had MCL1 robustly developed leukemia. So they got blood cancers and they eventually had to be sacrificed and died. But the cells that lacked MCL1 in red did not die, and this is up to 100 days. So that told us that when you have a blood cancer or leukemia where there's no MCL1, then it is unable to divide and proliferate in the body. So this is an encouraging validation in an in vivo context for the work, because it told us, like, within a mouse that MCL1 deleted cancers struggle to proliferate, and we validated that the mice that got the cells that had MCL1, so in blue, indeed developed leukemia as seen by the WBC or white blood cell counts during when they were sacrificed, and the triple knockouts just did not. So in conclusion from the work, I hope what I've shown you today is that we've demonstrated an anti-proliferative and DNA damage effect of MCL1 deletion and pharmacologic inhibition across different cells and tissues. And we've also uncovered a selective sensitivity to microtubule targeting agents upon MCL1 deletion. And finally, it hits a pretty important point of consideration, which is like, how do you balance efficacy and toxicity? So on one hand, combining MCL1 inhibition with microtubule targeting agents represents an attractive avenue for cancer therapeutics, because you can kill more cancer that way. But on the other hand, as I explained, you also have to be aware of the toxicity issues that can happen when you do that. So that concludes the scientific portion of my talk. I can quickly cover some aspects of career planning or some lessons I've learned along the way during my PhD, and then I would be happy to take some questions. Before that, I'd like to thank my lab. So as you know, like, science is a team effort. So there are a lot of people that are essential for the work that are in the slide here. And yeah, so with that, talk a little bit about career planning. So here, at least in the United States, as I've seen, once you get a PhD, you have lots of options. It's what traditionally used to be mostly either academia or academic positions, or pharmaceutical industry. Now you do definitely have a lot more options, such as, you could go into public health research, you can go into science communication. Of course, you can still do biotech industry or academic research, or even science policy. So the options are really limitless. So I would encourage students to not to keep an open mind as to what you want to do as you progress through your career. Always be kind of exploring your options, and do what feels right for you. And so I'll just conclude with some of these slides of some of the lessons I've learned along the way. So number one is, you have to embrace the unknown. Like, you have to step out of your comfort zone. Don't be afraid. A lot of times in research, as you'll find, if some of you go on to do your master's and your PhD, a lot of it is going to be unknown, and you'll feel uncomfortable. But it's okay. Like, you have to embrace that, and that's how new discoveries and new findings are made. And always have an attitude to learning. Like, nobody knows everything. You have to always strive to learn something new every week, every day, and be curious. Have a very strong bias for action. Don't be biased to different theories, but always take action. Dream big. You don't think something's impossible just because it looks hard. Just go for it and, try it. And if it still doesn't work out, it's fine. And, it's a team sport, so develop strong interpersonal skills, and good working relationships with your boss, your lab mates, and other people along the way. And yeah, it's important to find inner meaning and purpose in your work. So like, on one level, it's like the research and like, what are the, implications of that, but also like, you have to find joy in what you do. So always think about the bigger picture of how, the work or the research you do fits into your whole life in general. Yeah, with that, I'd like to thank everyone for listening to me speak, and can take some questions now.
We have got some questions here.
Okay, yeah. Okay, so let me see, I'm scrolling up. Yeah, okay. Yeah. All right, so first question is, how does MCL1 contribute to the regulation of apoptosis? So as I was saying, this protein, when it's there in the mitochondrial membrane, it prevents the cells from dying. So this way, the more of the protein you have, the less cells will die. And therefore, in cancer, they love to express this protein to high levels, because that way they can survive and contribute to the growth of the tumor. Someone asked about the ink of the octopus as an antidote to cure cancer. I'm not sure. I haven't really looked into that, so I can't answer that well. Someone asked, how does the expression of MCL1 vary across different cells and tissues? That's a great question. Yeah, so there's a lot of MCL1 in the organs. Like it's expressed in mostly all the tissues, but it's definitely highly expressed in the thymus, in the small intestine, particularly in blood cells as well, in white blood cells, in the liver, for example. So overall, all around, it's expressed, but with particularly highly in the tissues I just mentioned. What is the pathway of MCL1 and its main function? Its main function is regulating apoptosis, which I talked about. And then in the previous slide that I showed in my background, I also showed some other functions relating to mitochondrial energetics, fatty acid oxidation, blood cell development, etc. What are microtubule targeting agents? So microtubules are structures in the cell that help a cell divide. It's kind of like if you imagine your house or your building or a big building, let's say a commercial building, there's a foundation, right? They're like pillars. So microtubules are kind of like pillars in the cell. So there are compounds, chemical compounds that were developed to target these pillars so that when you take down the pillars, this building falls, right? So similarly, like when you target microtubules in the cell cannot divide and eventually dies. Okay, then billions of dollars have been invested in cancer research throughout the world, but still the cancer treatment is very expensive. Why? Do you think that after 5 years or soon, the cancer drug will be available for all in pharmacy within the budget of the government? Very good question. So this is actually a big problem in the US. Like cancer treatment is expensive anywhere in the world, but especially in the US, it is off the charts expensive, just drugs in general. So I'll give you an example very recently of this. So when I was little, I had asthma. So I used to have an inhaler with me at all times. And when I came to the US, I also brought it with me from India. And I didn't have asthma for many, many years. And then this summer, I had a little bit of it. So I needed the inhaler. And I saw the price, right, on the inhaler that I bought from India. And it was like, 300 rupees. The same inhaler here, if you want to buy it, is $327. The same one, exact same company. So that's like, that's basically the currency exchange rate fold. So that's 80-fold higher. And this is true also for cancer treatment. Why is it expensive? So it's expensive because, a lot of companies, biotech companies are trying to develop therapies. And, most of the stuff that 95% of the drugs that they try to bring to clinic don't work out. So they take a loss on the R&D research side of that for 95% of the cases. So the 5% winners, let's say, when it does work out, they have to kind of make their money back. So that's the way the biotech industry works. Otherwise, if they don't make that money back, there would be no research and innovation in the industry. So it would be harder for the advancements in clinic if there was no reward cycle for them. But the hope or the good news for us as like patients or for the people is that after, let's say 10 years, 15 years, a lot of these drugs become generic or the patent life expires. So every company can make them and then the price comes down. And this is like taking paracetamol, like when you have a fever, it's not expensive anymore. But when it was first, discovered many decades ago, it was, the people who were alive then had to pay a higher price. Now it's like cheap. So, with more therapies that come out in 15 years or so, they will get cheaper. But in the short term, they are expensive because there's no good solution. What is the success rate of gene therapy in cancer treatment? It's getting a lot better. It used to be hard, but there are lots of companies working on different gene therapy platforms. It's very specific to the disease though, like it's very personalized. So it depends on the cancer type, but there's currently a lot of promise in gene therapy.
I think now we have come to this end of this session. I want to thank you for such an informative and engaging talk. Your knowledge and expertise really helped us to learn. And apart from opening career opportunities in this field, an understanding of cancer makes people better able to support those with the disease, as well as help people reduce risk of potentially prevent cancer. As in India itself, it is the rate of cancer is 100 per lakh. That is 1 in 9 people are likely to develop cancer. I would also like to thank you for encouraging the students to explore new dimensions in their career path. Thank you, sir.
Yeah, it was my pleasure. Thank you for having me.
You're most welcome. And with this, we have come to the end of this first day of this 2-day online workshop. Feedback link will be shared in the Facebook, in the WhatsApp group. So I would like to request everyone to fill the form and hope to meet you tomorrow. The link for which will be shared in the WhatsApp group again. Thank you. Namaskar. Also, sir has given the email. If anyone has any questions, they can contact him. He has given his email in the chat box.